Assalamualaikum and a very good afternoon to all of you. Welcome to INBIOSIS APBioNETTALKS webinar lecture series 1 2022. I am Sarahani, your moderator for today's webinar. I hope the video that we played just now has provided you with some background of INBIOSIS. So for those who are interested to collaborate with us, you are most welcome to do so. Feel free to browse INBIOSIS website and social media for the latest updates and info. I feel so grateful today that all of us are granted good health and time to attend our webinar series. For your information, this is our first INBIOSIS APBioNETTALKS webinar lecture series for this year, 2022. INBIOSIS has also lined up a series of webinars to come, and I hope all of you will join us again in the future. INBIOSIS webinar series is established to provide a knowledge and research experience sharing platform to ensure we are still connected with each other and stay motivated in our research despite this sudden pandemic. Now, I would like to introduce our honorable speaker for today, Dr. Prashanth N Suravajhala from Amrita School of Biotechnology, Amrita University, Kerala, India. And allow me to share with you Dr. Prashanth's research background. He obtained a PhD in systems biology from Aalborg University, Denmark, and Dr. Prashanth then went to gain more than 7 years of postdoctoral experience across 4 different laboratories. He worked for Birla Institute of Scientific Research, Jaipur, on his return to India, where he successfully led 3 projects in the area of systems genomics of rare diseases and next-generation sequencing analysis of cancers and diabetes. His interests exploring the known and unknown regions in the human genome, primarily working on the top-down systems biology approaches of hypothetical proteins. Over the last 8 years, he has developed an interest in long non-coding RNAs in humans and elucidating the mechanisms underpinning small molecular interactions through clinical exomes. His group has benchmarked pipelines and developed methods for systems genomic integration. He is also an associate editor of Frontiers in Genetics or Systems Biology, Bioprotocol, Biomolecules, and also BMC Medical Genomics. Okay, now let us welcome Dr. Prashanth N Suravajhala to share his research finding. The floor is yours, Dr. Prashanth.
Thank you. Thank you so much, Sarahani, for this wonderful introduction. Thank you so much, for taking my memories back by keeping my photograph pretty young. I'm becoming a bit older. I would say when I say older, the photograph that was inserted was probably when I was 26, I believe. Now I'm 43. So it's almost 15 years behind. And that was a wonderful photograph. Thank you so much. So I hope you could see my screen, and I'm putting it in full screen mode. Can you all say yes if my screen is visible? Yeah.
Yes.
Yeah. Thank you. And I'm sorry for a certain background noise. In fact, I'm in the 3rd floor of my university campus where there are classrooms where there is a break actually. So I hope that this will not deter your interest in this. So. I also thank Zeti for her kind introduction to this wonderful university. So in the next 43 minutes or so, I'll take you to this wonderful world of rare disease genomics. I've been privileged to have analyzed a lot of rare diseases, studying them, learned some of them, and yet there are certain challenges and myths that one could really take forward. So I come from this beautiful lush green campus from southwest of India. This is where the state of Kerala is. And our university is ranked the top university in India. And it's among the top 100 universities in the world. And the best and the only institute of eminence in terms of the private institutes in India. And ours is headed by Her Excellency, the greatest humanitarian, Revered Mata Amrita, who has instigated these campuses in 2004, where our Amrita School of Biotechnology is led by Dr. Vipin Nair, where we have about 40 plus academic staff and over 600 students only in biotechnology, and about 30 plus PhD fellows and several other faculty associates and postdocs. So this is going to be the outline of my particular talk. I'll start with some kind of teasers somewhere in between. And I'll try to convince you that, yes, there are certain rare diseases that are not really rare. That is always at the helm of the definition point of view. And yes, rare diseases are not only diagnosable, but also treatable right from the prognostic phase. And I'll take a lot of 50% of my talk on the case studies, and the rest of the talk would rely something on the Acmg Guidelines, which forms the basis. Acmg is American Center for Medical Genomics and Genetics, and some of the epigenetics myths and challenges, and taking the long non-coding RNAs as drivers. I'll try to put this. So how do we define a rare disease? And this is still a kind of a burgeoning problem. If you really look into the incidences and the prevalences that are happening over the last, I would say, 2 decades, I think the rarity is not completely defined properly. That's certainly a kind of a very unfortunate thing. So, for example, if you take in US, 200,000 people in the US have been afflicted with some of the rare diseases. That indicates that about 6.4 people in every 10,000 people are afflicted with rare disease in the US. If you really take in Japan, you've got about 0.4%, taking the 50,000 prevalent cases in Japan, and 5 per 10,000 in the European Union, and 1 per 10,000 in Taiwan. And in Malaysia, unfortunately, although there is a rare disease consortium, that was there over the last 5 years, which I understand, the accurate definition is not there. So they take a lot of rare diseases, keeping in view of lysosomal storage diseases as a rare disease, where the incidence is 1 in 231,904 live births. So whereas in India has a lot of rare diseases, being such a big country. You have a big population, and about 7 to 8% population are tagged as rare diseases. And among the so-called 6,000 rare diseases, 85% of the rare diseases are only about a handful of rare diseases, about 100. The rest are all different rare diseases. So that vexes a kind of a problem that rare diseases are rare, but if given a kind of a proper attention, they can be treatable, which I'll try to convince you in the next few minutes. So how on earth we can come up with these particular rare diseasomic approaches? So we know that there are contexts in the systems. If you take a human as an organismal system, or if you take, say, any organism like eutherian or any living organism as a kind of a system, contexts are nothing but ohms. Ohms are nothing but many. Okay. So if you take a plant, you might herald a kind of a plant with something like stress traits, the UTRs, the genes, and all this stuff. The animals, you talk about the feed traits and efficiency. Something like in bacteria, you talk whether or not the bacteria is a monoisolate or a multi-isolate, or whether or not it's non-pathogenic or pathogenic, host-specific, host non-specific, and all those kind of things, so the gram-positive or gram-negative. Whereas in humans, although there are such kind of similar traits that we always talk about, similar variables that we talk about, we always talk about the influence of the genes on how the environment is, or how those particular phenotype is being influenced by that particular environment, how the particular disease is being influenced by the environment through the genes. So this is what we call it as epigenetics. Okay, so epigenetics plays an absolutely key role for this, and that's where you can say that there are a lot of true positives, a lot of false positives, a lot of true negatives and false negatives that are associated in describing the accuracy of a disease. So that begs a kind of a big question, when it comes to the rare diseases. So when a patient or a family member takes a person to the doctor at a very immediate effect looks at the disease phenotype, then he or she would really probably ask a lot of questions. What's the diagnosis? Was the prognosis done? Whether or not it's identified properly or misidentified? And if the patient is absolutely in a kind of in a different situation, you may say that maybe the person might be associated with a mitochondrial disease, and whether or not if any of these particular alleles are coming from autosomal dominant disease, like something like you might get a gene that is afflicted from either the parent, the allelic variation coming from either the parent or the mother or father, okay, largely coming from those particular things. And many of these particular mutations are largely, I would say, heterozygous, although homozygous mutations are largely associated with a few rare diseases. And most importantly, whenever you say it's a rare disease, you always talk about monogenic condition, which means usually only one gene is usually causing that particular disease. And then we go to the treatment. There is absolutely very costly treatment process. A lack of consensus. When you take one person to one doctor, maybe the doctor would probably prescribe these particular genetic tests. The genetic tests might really be misdiagnosed sometimes, and maybe the bioinformaticist or maybe the next generation sequencing analyst who does that comes in a different way. So a lot of things come into picture. And there could also be a lack of availability of something what we call it as a pedigree correlation, whether or not if a person is there, can we get a kind of a sample from their forefathers, or at least from their particular parents, or at least from their quad, which means unaffected father, unaffected probably mother, and at the same time maybe unaffected or affected sibling. So this kind of quad-based analysis comes into question. And that's where we could probably bridge the gap. We need consensus from geneticists. We need consensus from what I call a clinician. We need a consensus from probably the person who does the analysis, whom we call it as systems geneticists like me. And we also probably need whether or not the information is really coming correctly from the pedigree. So moving into the holy grail, so we all pretty well know, from bioinformatics or systems genetics perspective, sequence predicts structure, structure predicts function is what we know as a kind of an orderliness for defining the function of the protein. But many a time, the sequence wouldn't fold properly. So when you have a sequence like this, a simple sequence like this, if it makes rigorous folds, that indicates that those folds would have a lot of pockets, a lot of groups. That those groups would allow the other ligand to bind to that. Okay, but in nature, unfortunately, not all sequences are foldable, which means not all sequences fold impromptu properly. Okay, there might be sequences that might be semi-structured, which means you really don't get a proper structure for those particular sequences, and those particular structures are called as disorderliness or disordered structures, and that would have no function at all. So you don't have any kind of function for those particular things. So why do you get this? Because as I was telling you, there's always an epigenomics, always associated with this, and maybe that single mutation might really or miserably or unfortunately cause that particular mutation. So what you say is what you get. This is what we call it as the Wiswick principle. If some of you is from a computational background, so you get a sequence, structure, function, all in proper order. Probably you get a normal, healthy person or a healthy persona. But if you have a sequence where you don't have a proper structure, or maybe the structure is not in a good shape, maybe you might have a kind of an impromptu disorderliness. That disorderliness might have a lot of environmental influence. And that's where these epigenetic changes, influence of the histone modifications, and whether or not the chromatin elements, open chromatin elements are modeled properly, and at the same time that genotype phenotype correlation would really come. And that there lies a kind of a chance that there could be a chance that there could be non-coding RNAs influencing these particular mutations, because genetic variation is best seen in the non-coding RNAs when we talk about the coding or the non-coding mutations. So when we have such genetic basis, to be prognosed or to be taken to the next level, we should always talk about the heritable mutations, whether or not the mutation is really coming from either parent or maybe from the ancestry level. So it's always nice if that is the case. There could always be, some things like, consanguinity as well. Sometimes, many consanguineous marriages, herald a lot of, homozygosity, and that results in a lot of, rare diseases as well. So when you have such individual variants, so we are talking at the variants at the individual level or mutations at the individual level, which we call them as a single nucleotide variants. So calling or getting those single nucleotide variants from the next generation sequencing is very, very important to understand the diseasomics of a rare disease. So if we have a rare disease, and if you really don't know what that particular rare disease is, if you really want to know what is that particular condition, that you really want to get hold of, it's always nice, to get, some kind of, good pipeline that will allow us to validate or identify those particular mutations. So the path to discovery for rarity, can be a multifold approach. You can largely go for a whole exome sequencing or a whole genome sequencing like this. These two are the most important thing. And the whole exome sequencing or whole genome sequencing can largely be taken into consideration, taking a larger depth. So which means you chop up the sequences into similar fragments, maybe 50 bases to 150 bases or 500 bases max in case of a short read sequencing. And it's always advisable to have a wonderful depth of coverage, for those fragments. So if you have a larger depth of coverage, which means if each of those particular fragments are sequenced, say 50 times or at least 100 times, that indicates that the chance of identifying that particular variant in that particular individual is very high. Okay. And of course, that all depends on what kind of fragments that you're really looking. Maybe you can also look for a well-known rare disease where we can directly go for a targeted genome sequencing when we sequence only those particular genes or those particular regions of interest, and then you can take this to the next level. Okay, so these are all some of the challenges, and of course you need to validate it. Today we have wonderful approaches apart from the Sanger validation. We have got the Sequinome, we have got the Casper. Even we can even validate using the Maldiktov based approaches where we can identify that particular mutation of interest, identify using a simplest mutational spectrum or a capillary electrophoresis, and these and read those particular bases using those fluorescent tags using the Sanger chemistry. So these are all very must, and then you'll be able to find what are those mutations that are pathogenic or that are malignant or that are disease associated, and largely many of these whole exome sequencing based approaches have been used as a discovery tool to identify these rare diseases. Albeit the fact that there is a fallacy, because in a whole exome sequencing you only chop up the exons. So from the previous slide I was telling you here, you can only chop up the exons here. So you can only identify the mutation sitting only in the whole exome sequences. Okay. Albeit the fact that there might be certain mutations that could be intergenic, that could be maybe cis region, or that might be at the trans region. Okay, they could be in a long intergenic conditions as well, where they could sit somewhere in between the intron exon boundaries as well. Where you could probably identify those mutations. But the real challenge is, the real fallacy is probably you might miss a kind of a rare genetic variation that is affected from a non-coding RNA. So that could really be a kind of a challenge. That could be a kind of a deterrence for you to identify a kind of a mutation. So to overcome this, to overcome that particular thing, probably you can do it again a targeted genome approach, or you can use some well-known instruments, where you can use 10x genomics, where you initially do the long read and then go for a short read sequencing with an Illumina read. You can identify, absolutely good number of mutations using different platforms. The latest being Illumina NovaSeq, NextSeq 2500, and all those wonderful instruments. However, looking into the epigenetic spectrum, looking into whether or not there is a regulatory aspect associated with this. One of the other challenges that one would really look into is whether or not any chromatin region is undergoing any kind of changes like acetylation, methylation, okay, or methylated events, within those particular chromatin elements, where a large amount of histones are modified, thereby the downstream pathways where the gene is really transcribed to mRNA and finally mRNA is translated to protein is being affected. So that's where this ChIP-seq, the chromatin immunoprecipitation sequencing assays have really come into picture. They are absolutely great models where you can identify what are the transcription factor binding sites, what are those histone modifications, whether or not there's a good compatibility, and then you can even compare it with the regulatory genome approach using the methylation spectrum, and all those wonderful things can be used. But despite you using so many tools, investing this much money, but if you're not able to identify the mutations, the accurate amount of mutations, or maybe the variants in those particular regions, then you're at a huge loss. So keeping in view of this, since 2012, American College of Medical Genetics and Genomics, the Acmg Guidelines have been in practice. They put a lot of best practices and guidelines, which they promulgated in 2012. So they gave a wonderful classification saying that if there is a mutation in this particular region, then you may say that this particular mutation might be pathogenic very strong. Or maybe these are the things, then it might be a benign mutation. These are the things, then these are all the things. So likewise, not only the ACMG, but also Association for Molecular Pathology, and also in UK you have got Association for Clinical Genomic Sciences, you have got Association for Clinical Genomic Sciences for Australia, and for individual countries you have got different things. And for Singapore there is one, and for India, of course, we also follow something called the ACMG Guidelines as a gold standard. And then you come up with an evidence-based classification whether or not a variant is very strong, or moderate, or simply supportive, and what are the types of data that you use. You will use whether or not the data is coming from a large population or from a simplest predictive analytics, which means some data only say out of 100 exomes, maybe only 20 exomes might be sequenced. The other 80 exomes are not done, but you come up with a prediction, taking these 20 exomic chunks, and the functional annotation and all those kind of things, and then you classify whether or not that particular variant is pathogenic in nature. You might say that if at all, if these particular classification fails at some point of time, if you are in a doubt, you may always say that it's just likely pathogenic, or maybe benign, or maybe causal or bona fide. So the bottom line here is you need to keep less discordance. Okay, you need to use something what we call it as keep it concise and simple, which we call it as a KICS rule, which applies largely with such kind of variant calling approaches. And of course, you use various tools like dbSNP, dbBar, CleanBar, and all those particular things for keeping in view of brevity of time. I'll not get much into these kind of things. And then you go for various databases like the GERB, the Genomic, the Genome in Bottle, the CADD. You can also use the Indian genome databases like the IndexDB, the SAGE, the Indigen, and several other databases that are largely used for the rare diseases as well. And then you might come up with a question: what are the kind of mutations that need to be looked into? Prash, you said that you have whole exome sequencing data, which you say that it's a gold standard. But what if you have mutations that are only associated with missense, because missense mutations by any chance you have almost 70% of the mutations are missense. Nonsense mutations are those mutations where you have a change in the amino acid when the mutation changes. And you might even have the stop loss, stop gain mutation, or even multi-exon deletions or single exon mutations. But unfortunately, many of the mutations associated with the rare diseases are all the variants of unknown significance, which we call it as VOUS or VUS. So the variants of unknown significance have really been dealt over the years, and they have been really been playing a huge role. So I'll just give you a kind of a classical example for this. Okay. If you see the cancer picture, okay, you see the BRCA1 and BRCA2 MLH1 as a DNA mismatch repair that is largely associated with HNPCC, which is a colorectal cancer, homonon-polypolysis colorectal cancer, which is largely seen in Malaysia and Singapore as well. MSH2 and CFTR. If you see these 5 mutations or 5 genes, about 50 missense mutations are there, in them. You see 50 missense mutations only within these 5 genes that are largely associated with this. Then how do you identify them? You can identify, of course, taking the whole exome sequencing. You have a capture kit, taking the entire 1.8% of the coding chance. Whether you can take a whole exome sequencing kit with 34.1 MB, 37.6, or 47.9, and the latest is 68 MB region as well, which also coalesces some intron exon boundaries as well. But keeping in view of such pleiotropic nature, which means one gene need not really cause only one disease, but one gene might be associated with several diseases as well. So if you take BRCA2 is a well-known breast cancer mutation, but it also causes a lot of mutations in cancer prostate. MLH1, we know that MLH1 is a DNA mismatch repair protein. It not only does a DNA mismatch repair activity, but it's also known for hormone polyposis colorectal cancer. Okay. So we have such kind of challenges, and that's where these ACMG guidelines are useful. That will allow us to indicate taking the computational data, the functional data, the segregation data, probably the de novo data, the allelic data, and other things, and then prioritize what are those particular variants, whether or not the variant is strong or very strong, or simply supportive, or if it is simply benign in the sense, it's not causing a disease, it just, there's a mutation, which fails in causing a disease, okay? And then, we can classify null variants. Null variants are those particular variants. Now, this is a typical, pathogenic variant, very strong mutation, you see. Null variants are absolutely benign, which means they do not cause disease, but they're heterozygous. It is a very strong pathogenic variant because the phenotype is absolutely associated with the autosomal dominance region, which is a mutation is really coming, from one of those particular parents that is largely associated with hypertrophic cardiomyopathy. Okay? And this is largely seen in CFTR gene. This is very unfortunate. So that you have a mutation, and yes, you're calling it as very pathogenic, and yet it is a benign. Okay, then you need such null variant classification as well, and that's where these 3 C's in practice should really come into picture. So the 3 C's are nothing but you need once you identify a variant or detect a variant, you get recommendations from clinicians. You ask whether or not any functional assays or genetic tests have been performed. You reconfirm twice or thrice. Sit with a clinician, sit with a geneticist, sit with a NGS expert, and identify whether or not those particular variants are so and so. And for that, we need a 3C principle, which I call it as consensus, collaborate, and converge. You cannot sit idly. I mean, a clinician cannot say that, yes, I'm a doctor and I could probably come up with a diagnosis. He or she has to sit with a geneticist. He or she has to sit with a NGS expert and also with a family member during the counseling session and come up with those particular things. So the first part of my particular talk here is that there are ACMG guidelines, which forms a large amount of framework. The fallacy, however, is that there should be an accurate development of quantitative measures that needs to be practiced. There must be 3 C's in practice. There must be a greater collaboration and convergence between geneticists and clinicians. And then I have also given you a classification that you don't identify or validate, taking the GWAS data, the genome-wide association data, but you also take, the further downstream whole exome or clinical exome, or maybe, the downstream validation, like, targeted genome sequencing. And of course, the epigenetics is, a kind of a key factor. So moving further, when the ACMG guidelines were used, ACMG guidelines also has a kind of a fallacy, because that fallacy is that, that fallacy, as I was telling you, that you don't have that quantitative measure. So then came this Sherlock Criteria, which is a kind of a semi-quantitative measure, but you can also identify whether or not the locus standi for interpreting that particular mutation is absolutely impromptu associated with a disease or not. Whether or not you say that that particular variant is absolutely causing a disease, whether or not you say that that particular disease is already established by ACMG AMP guidelines, Association of Molecular Pathology, and American College for Medical Genetics and Genomics framework or not. And that's how this looks like. You classify that, you come up with the population data, you take up the clinical data, you take up the ACMG functional data, and then infer this particular pathogenic state and come up with a greater evidence where this Sherlock et al. In 2017, they recently framed this 5 years ago. But when we were annotating a lot of variants, a lot of mutations coming by and large from a lot of diseases, a lot of rare diseases, we don't even have sometimes the data coming from the parents as well. We don't have the parent sample as well, because sometimes a child is born and the mother is gone, a mother is lost, and we don't have sometimes those kind of things. And that's where a lot of mutations pop up in a small region like this, a small region which we call it as this particular condition in genetics is called as a linkage disequilibrium, which means a non-random allelic association is associated, which means you really don't know which SNP is causal to that particular disease phenotype. So a small 2000 base pair region between SLC4A4 and NPFFR2 in chromosome 6 is largely within a matter of 2000 bases, you find a lot of what you call mutations, over 1200 mutations, but you really don't know what are those particular pathogenic mutations among them. So then you can identify the challenge here is you can identify whether or not that mutation is heritable, whether or not that mutation is integrated with Bayesian statistics, crude statistics, whether or not it's heterozygous. In case of some eutherians where we have taken the milk data, we have taken whether or not it's a somatic cell count, whether or not any variation is sitting from the long non-coding RNA chunks from the whole genome sequencing, and that's how we can infer whether or not a bona fidelity of that particular mutation can be taken up. Okay? And then we look into the genome browsers like this, we go deeper and we see if there are any open chromatin elements or functional data that is associated, or any regulatory regions or regulatory elements associated within the vicinity of that particular mutation, and take those particular mutations and identify what are those particular mutations sitting across those 4 classifiers. So here we have these circles. The yellow one is the circle which is seen across all the 4 conditions, all the 4 classifiers, which we call it as functional annotation, disease condition, and pathogenic variant, and all those kind of things. And then we say that this particular mutation is diseased or causal. This particular mutation is really causing a disease, because this particular mutation is sitting largely in all the 4 conditions, in all the 4 pies. And then I may say that this is a disease condition. And then we look into the pathways, we look into the disease phenotype, we look into the patient statistics, we sit with the clinicians, and then we come up with a standard inference. So what we have done over the years is, over the last 10 years is, we have prioritized SNPs. Out of so many SNPs, we can say that yes, since the genetic variation is sitting largely in the non-coding chunks, we ask whether or not that genetic variation is sitting in the non-coding RNAs, whether or not that genetic variation is sitting in the functional annotation, whether or not that genetic variation is sitting or associated with an open chromatin element, whether or not that genetic variation is sitting largely in the methylation spectrum. Then we may say that that particular variation is largely influenced by epigenetics, and then we come up with a disease condition. Okay, so I'm going to take you the next 25 minutes or so, to these wonderful case studies. Okay, where we have done it. So this was a case study which we did in 2016. India is a diabetic capital. Over 26% of the official population are diabetics, and many of these particular, since population is diabetic, many of them are also associated with something called lipodystrophy, and many of these lipodystrophic genes are largely lamins, and over 40 genetic tests are already there, for identifying whether or not the mutation is sitting in the lamin gene or not. But there are also other genes by the advent of pleiotropy, which I was telling you. You find a lot of genes like PIPA gamma, the Kevin genes, even the monogenic insulin-resistant genes like PIN. All those particular genes are also there, and a lot of 49 risk factors are already there, associated with these particular diseases. That begs us a question: Are we really obligated to do only a lamin sequencing, something like LMNA sequencing, or can we do something like a sequencing that is largely the whole exome or the targeted genome sequencing? And then we can say that we can go for those particular things, but unfortunately, not all genetic tests are associated in that particular place or city For example, even in India, we don't have that particular test for that. If that particular disease is rare, we only have tests coming from only state of Iowa, which is there in US, which is in University of Iowa Diagnostic Laboratories, which is very, very unfortunate. And then we had to look into these particular genes. What are those particular genes? What are the genes that are physically interacting? Then when we can say that those particular genes are physically interacting, that are largely associated with the diabetic conditions, and then come to a kind of a conclusion. So a quick summary of the case one from the clinical case history. Perhaps there are really no verified genetic tests for such LDs, okay, for lipodystrophies, but we have to rely upon the protein-protein interaction networks, do a pull-down assay, do a PMS2 based assays, okay, and all those things, and then we have to take a gene called PPA gamma, which is largely associated with diabetes, take it as a bait and come up with a simplest pull-down assay, GCK as a prey, and then we have to again sit with the clinician and come up with these conditions. Okay. So the second case study which I really wanted to convince you is not every time the genetic variation would be seen in the coding chunks. You would also find the genetic variation sitting in the non-coding chunks, as I was telling you. You might have a non-coding RNA sitting like this, where non-coding RNA, long non-coding RNA could be bidirectional like this. It could be sitting at the sense strand, which is at the cis strand. It could be sitting at the antisense, which is at the trans strand. It could be intronic, which means it could be sitting between the two exons, or it could even be sitting between the two important genes, or it could even be the intergenic regions as well. So, and then we know that the genetic variation is sitting in the long non-coding RNAs, okay? And then we come up with a kind of a conclusion saying that yes, the long non-coding RNAs derive the methylation patterns, derive the epigenetic factors, derive the epigenetic activation, derive the kind of conditions that we are really looking into. So these modulations would really allow us to identify what are those particular rare variants for those particular rare phenotype that you are talking about. So for that, we came up with a wonderful thing. We asked, where with all and why with all, we find these particular long non-coding RNAs. Why is that, we find those long non-coding RNAs? So long non-coding RNAs by definition are more than 200 bases in length. They are non-coding, but they have coding potential. They might be coming from 5 of the untranslated reading frames of the 6 reading frames, okay? Because you have 6 reading frames from the code being triplet. The other 5 reading frames might be untranslated, and those particular untranslated reading frames could emerge as long non-coding RNAs. But before a long non-coding RNA is formed, they are nothing but the hypothetical proteins, okay? They are probably misannotated, okay? And then annotators or curators like me can identify such known unknown regions. A known unknown is nothing but a known gene, but an unknown function. Okay? Those particular things we did that with using an example called Link 00208 as a kind of a case study. And we found that this particular long non-coding RNA is influencing a largely associated with 3 types of diabetic conditions: type 1 diabetes, obesity, and type 2 diabetes. You see 3 different conditions here, okay, wherein a lot of genes are interacting with each other in 3 such particular conditions, and you don't have a common genes between them, but you have a common genes between obesity and type 2, and of course type 1 and obesity. Okay, so you've got almost 12 genes because type 2 diabetes is always obesity, and among them we find this largely the long non-coding RNA, which is influencing that rare epigenetic events associated with diabetes as well. I mean, this is largely again associated with not only diabetes but downstream conditions like the lipodystrophies, okay, all the monogenic disorders associated with diabetics, which are absolutely rare. So the quick summary of case 2 here is the known unknowns could be non-coding RNAs. Many of these particular non-coding RNAs emerge from different diseases, and many of these long non-coding RNAs have special place for regulation. Hypothetical proteins could serve as predecessors for long non-coding RNAs, and of course the long non-coding RNAs have some kind of coding potential. The third case study is from our another collaborator, Dr. Sudhir Mehta, which is what we call it as aplastic anemia, which is a rare hypocellular bone marrow disease, where a lack of hematopoiesis leads to decrease in reticulin condition, and as a result, the patient is very deficient, with the blood levels, and he needs, kind of a blood transfusion every time, or he might need a kind of an immunosuppressant. We know that telomerase reverse transcriptase gene, which is a third gene, is a well-known gene, but that's the only monogenic condition for this aplastic anemia. But we also found some other genes, affecting Indian phenotype here. So when we did a whole exome sequencing, we not only found the third mutation, but we also found the PGA and IFNG mutations, which are missense and stop codon mutations. Many of these particular mutations are germline mutations, which means those particular mutations, from our predictive analytics, come from the hereditary lineage that has been recently discovered, from the UK and the US consortium. Unfortunately, in India, we don't have such particular thing, and this is our first case in India where such aplastic anemia with over 60 or 50 samples have been identified for whole exome sequencing, and we found those particular mutations. So we may say that the cyclosporine treatment or the blood transfusion treatment is also given for those particular patients who might need these kind of things. And of course, aplastic anemia sets a precedent, for such immunosuppressants. The fourth condition is a very sad thing, which is something very close to my heart, which we have been doing it over the last 1.5 to 2 years with our clinician, Dr. Naresh Tayade. There is a patient named a small baby aged DB, okay, named DB, who came, 8 month, who was an 8-month-old patient diagnosed as congenital bilateral cataract. No sample from the parents. The parents were not really willing to give the sample, and the baby has a development delay. The baby is about 3.5 years old. There is something early onset intrauterine growth retardation that was seen, when the CT scans were taken. The baby was born, premature, and he was kept in an ICU for about 8 days. And the examination showed that there is absolutely no bilateral cataract, leading to intraocular calcification, where a lot of calcified tissues do exist in the eyes, making the child cataract. And then we identified a lot of non-coding RNA platelets like this. A lot of, first we asked if there is any karyotypic dysregulation. There is no karyotypic dysregulation. We found a lot of non-coding mutations. We could ask our service provider to chop off those particular intron exonic boundaries and look into those particular mutations over there and come up with those deleterious mutations. We found those particular mutations, largely coming from the intronic boundaries like this. And some of the mutations are also the non-synonymous mutations as well. But these 7 mutations, as you could see, okay, and the coding mutations are always deleterious. Okay, although the deleterious mutations are low confidence, the tolerated non-coding mutations, as you could see, they are absolutely, from the SIFT database indicates that there are mutations coming from the non-coding chunks. Okay, so the congenital bilateral cataract is a rare disease. It's phenomenal to various countries, but in India, it is first exploited. It is largely associated with phosphodiesterase gene. And Indian phenotype has about 6 mutations across 4 genes. Unfortunately, no carrier mistyping. And our phenolizer data indicates that rare disease conditions do exist, but we're still trying to figure out whether or not these 6 mutations are really impromptu, so that we can come up and check with other clinicians and genetic counselors and develop a kind of a proper panel. So the final case study is something about a rare phenotype, which we have published quite a lot, thanks to Dr. Praveen Mathur, a clinical collaborator. Is a doyen of this congenital pouch colony, has done more than 800 surgeries so far. The child is born with a dilated pouch, and the urogenital system is pretty bad. It comes with a dilated pouch. The excretory system is very bad, something like this, and the child needs to be operated very soon. And it's a rare condition. It's largely seen in males, but also females are also seen. A large amount of disease is seen in consanguineous population, and we initially did 64 exomes, and then we also did something what we call as a trio analysis and also the quad analysis. Trio is nothing but an index case, the congenital case, sequencing along with the mother and father, and also an unaffected sibling as well. Okay, and the oldest child at that point of time when we got was an 18-year-old child, with this congenital pouch clone. He was still a child because he's still less than 18 years old. Okay. And then we found a lot of mutations. We published this in scientific reports and other journals in biomolecules and other things. We found a lot of heterozygous mutations. A lot of mutations were afflicted from missense mutations like this. We also have some mutations largely coming from stop gain mutations, stop gain alterations. A lot of frame shift mutations associated with congenital pouch colon as well. But assuming that frame shift mutations do exist, then we typically found a very serendipitous result where we identified a long non-coding RNA gene harboring a mutation, and that long non-coding RNA mutation was seen in a gene called link EPB41-1, and we identified whether or not this long non-coding RNA KIF13A, which is another gene that is known to be causing, are interacting or not. So we did some bioinformatics heuristics and came up with a long non-coding RNA protein interaction data. And they used a wonderful technique called microscale thermophoresis, which is a biophysical instrument. The take-home message here is, in this wild type, you see all the biomolecules are poles apart when a thermal gradient is given, whereas here in a sigmoidal curve, you see all those biomolecules sticking to this particular line, indicating that these biomolecules are interacting. And these biomolecules are nothing but that long non-coding RNA and the KIF13A, the ligand and the protein biomolecules are sitting, just adjacent to these particular so-called, sigmoidal curve. So, but we didn't end up over there. We did further quad trio-based analysis. We checked for heterozygosity. We asked what are those particular mutations. We did a whole lot of things, and the paper has been communicated just now. And we did a lot of base studies, and this is the only trio-based analysis for such a large population for a rare disease in India. That was a huge cohort one way, and thanks to our collaborators, Dr. Krishna Mohan and Dr. Praveen Mathur, who made this cohort possible. And we published this in a decent impact factor, which was the first serendipitous study from the long non-coding RNA protein interactions associated with pouch colon, which was largely downloaded in this 6.3 impact factor paper. A quick summary of case 5 here is that, have you ever thought that we can find mutations from a whole exome sequence coming from long non-coding RNAs? No, that was certainly a kind of a myth buster. That was certainly a kind of a serendipitous study. So we found those particular mutations, and we checked using a microscale thermophoresis whether or not those particular genes are bound biophysically using that microscale thermophoresis instrument, which uses a temperature gradient, and that indicates whether or not those particular genes are indicatingly bound to each other. If those particular regions are bound, we may say that yes, there could be the mutations that might be bound to those regions where these particular genes are also bound. And we found these regulatory-based approaches, and we identified these particular challenges. So the final part of the talk here is, having done so many rare exomes, we have done about 200 rare exomes alone, and about I would say about 350 exome data that we have analyzed. We have a huge set of data which we consider as part 1, and we have data coming from the long non-coding RNAs as well as part 2 as well. And then, can we identify in the near future? Can we predict the state of the art of rarome? Can we identify that there could be some mutations, there could be variants that are largely coming from those rare genetic, allelic spectrum from these particular data? Can we apply machine learning heuristics? Can we apply, artificial intelligence into this particular thing? That is where we are coming, we are largely looking into. So the overall conclusions here is that the epigenetics is the key. Environment has got a lot of influence, and rare phenotypes here are certainly rare mutations. It is certainly treatable, if not inaccurately diagnosable. And there's a need for bringing those systems genomic approaches. Annotation and curation is very important. Non-coding RNAs mutations are also very important because genetic variation is best seen. We need those data coming from the metadata. We might also use the ChIP-seq data for identifying the histone modifications, so that we can better identify the regulatory role or we might even get to know whether or not they're interacting with any other partners. And most importantly, we need to bridge the gap between clinicians and geneticists. That those three C's, if you remember, consensus, okay, is very, very important. Collaborate is also, very much, important. And the third one is converge. We need to converge your ideas. You cannot say that I'm a doctor, I'm a geneticist, I'll keep my ideas only with me. You cannot say that. We have to, come together and join hands, towards this. And that's the reason why these another three C's are also very important: collaboration, convergence, and consistency for development of these things, especially post-COVID times. We are being deprived of a lot of data, a lot of man-hours is wasted. We humans, during this post-war times, we need to come together and at least be tad human in exchanging these wonderful ideas, so that we can do wonderful justice to the humanity. And we need to be good listeners. We need to come together and overcome all this kind of stress, and we need to maintain an adequate gender balance, ensure that women is also being empowered greatly, and we should also ensure that a lot of the leadership qualities stems from our individual backgrounds, so that we can overcome these two important syndromes called the imposture syndrome and the Dunning-Kruger syndrome. So when the virus came in 2020, 19 December, by the time in March and April when the lockdown was happening, we thought that by Indian summer comes, the virus would fade away with that particular summer. But you might be surprised that only during summers across the world you find these particular mutations, which means the mutations has understood the human intelligence. It has overcome the human intelligence. It has known, how the human is behaving. And when a small virus, which is invisible, can bring that kind of intelligence, why not we humans, can come together, for this? This is where, we all need to come together. And finally, I'm a part of these two wonderful call for submissions. You're most welcome to submit your topics to this. You can contact me at crash@biotechnology.org. These two things. There's another journal in biomolecules and this thing in Frontiers in Genetics. Two calls are open, and if you have papers, please contact me. And I'm also the founder of BioClues, India's largest bioinformatics society. We have been spearheading the bioinformatics community over the last 17.5 years, and we have been doing organizing a lot of events. We have a Women in Biology forum. We have bioinformatics for school children, several BioClues interest groups created through WhatsApp. We have an INBIOSIS Indian Conference on Bioinformatics 2022, this time in Guntur, in southern city of India, where Sir Richie Roberts, Nobel laureate for alternative splicing, has kindly consented to give a keynote. So please register for this. The news will be available somewhere in bioclues.org very soon. Okay. So I think I'll probably stop here and thank Professor Zeti, Sarahani, and Murni for the kind invitation, and Team UKB and APBioNETTALKS and everyone. I would like to thank our Guruji and Guruma, my spiritual gurus, and of course Gurus Mata Amrita and our head Dr. Vipin Nair, Dean ASBT. And all these works would not have been possible without my clinical collaborators, KM and PM, Krishna Mohan and Praveen Mathur, who are my mentors, who have given a lot of clinical samples for us. Sudhir Mehta, Sudhir Mehta, who is a well-known hematologist, who has given aplastic anemia samples. Naresh Tide, all those the DB cases that we have been analyzing. Obel, of course, with whom we have been collaborating a lot, and a lot of other players like this have been listed here. Come here, and of course my PhDs and postdocs. In fact, my rare disease student, PhD fellow, now is a postdoc. Sonal Gupta has done a lot of work on this. She was a wonderful, terrific PhD fellow. And we have Akshay, Anjali, and Gautam, who are currently working on these rare diseases, who are re-benchmarking the pipelines or reanalyzing these things, because we need to come up with the reproducibility thing. And all other my PhD fellows who are here, and my postdoc and other particular members who are put up here. I'd like to thank the funders, the Brain Pan, the Doctor's Diary, and the SERB-DS2 has given me another funding in the form of vitamin K. And I think I should probably thank the organizers here. And I look forward for the questions. Thank you so much.
Okay, thank you very much, Dr. Prashanth, for sharing your interesting study. So there are many case studies that have been shared, so we can see in terms of application in medical field, specifically in rare diseases. For those who are interested to ask question to Dr. Prashanth, please write in the chat box so I can read the questions for you. So I have already have a question here in the chat box by Manjula Tima. So the questions are these genomic variants are augmented with functional validations like transcriptomic data?
Yeah, I mean, that's indeed a very good question, Manjula. In fact, not necessarily, or I would say not really. If you find certain mutations, maybe you ask a service provider and say that maybe you want to chop off certain inferring boundaries as well, and then say that you'll find certain mutations from the non-coding chunks as well. And then you directly go for what we call it as a targeted genome sequencing, which means you go deeper and only sequence only those particular regions of interest. But sometimes it is still beginning to be understood, which we are also trying to analyze, that the genetic variation is best seen in the non-coding RNAs. So when you have the genetic variation seen in that particular way, you also expect that those particular genes could also be differentially expressed. So which means the mutation in which the particular gene is covered, okay, or the gene in which the mutation is covered, that gene, whether or not it is differentially expressed or not, you may try to analyze using RNA-seq, but you can do a simplest qRT-PCR for that. You can do a simplest mutational spectrum for that. Okay. And I don't think you have to shell out a huge money using RNA-seq.
Oh, okay, yes. All right. I think that answers Manjula's question, right? Okay, so I am quite eager to know actually what inspired you to initiate this research on rare diseases.
Sorry, what is, I'm sorry, Sarani, what did you say?
Yes.
Please come again.
I would like to know what inspired you to initiate the studies on these rare diseases. Do you have any do you have any collaborator that approached you on these rare diseases?
Yes. I mean, my first experience comes largely from, I would say, when I was doing my, this 8 years of postdoc. I had a large gap, not large gap, but 3 or 4 months gap moving from a PhD to postdoc. And what I was not trying to understand was, can we, come up with certain minor allele frequencies? Can we identify minor allele frequencies that are largely 0.01, which is a minor allele frequency, when we say minor allele frequency of 0.01, which is 1% of that particular allelic variation sitting in that particular population, which is afflicted with a rare disease. But again, you can even take an extremely rare condition where you can take 0.01% as well. But when we used to check the gene banks, there used to be a lot of rare diseases hovering between 0.01 and 0.05 as well. And those particular rare diseases were not classified. They didn't have, I mean, at that point of time in 2010 and 11, there was no ACMG gold standard guidelines framework. There was no Sherlock criteria and all those, kind of things. And then we were, working on a lot of other diseases like, colorectal cancer, working on diabetes and all those kind of things. All those diabetes associated diseases are all already there. So that was what, we tried to look into those particular conditions. We asked, what are those particular diseases that are largely, coming from what we call minor allele frequencies between 0.01 and 0.04. So I developed an interest in rare diseases during that particular time. And second thing was, many of my friends and my family members, they were not from the consanguineous family, but they were yet, with, I mean, they have, their children, with rare deformities. So I developed that kind of, interest towards that. and I put it as a kind of a practice in this kind of this. Thank you.
Okay, great answer. Okay, next we go to another question by Sugunakar. So the question is, linkage disequilibrium might be the key in establish the rare disease association with genotype. What is your say on this?
Yeah, that's what I was trying to tell Sibu. I think linkage disequilibrium is the main culprit. In genetics, it's certainly the kind of a culprit. If we tend to see a kind of a linkage disequilibrium, first, we'll ask whether or not we have find a mutation and we have found a gene responsible for that particular disease, which means we tend to come up with a monogenic condition or biogenic condition, which means we'll find two genes maybe sitting adjacent to each other. And what's the difference between those particular two conditions? Okay, what's the length between that? And then even from the omics point of view as well, even if we find to observe a lot of mutations between these two important genes, even if we find a lot of SNPs where we have the allelic dysvariation, even if we tend to observe that not all SNPs are really pathogenic in condition, then that is really a kind of a challenge. So we may have to maybe go for something called traceback mechanism, which means we identify the mutations in principle in that, and probably targeted genome sequencing approach, maybe looking into only those smaller KB region size, resequence those particular regions, and then overcome the challenge of identifying linkage disequilibrium. But this linkage disequilibrium is largely seen with only inexperienced condition, which means you'll only find one or two rare diseases with such LD conditions. You may not really find LD across all the rare diseases. So then you need bigger population size as well. Maybe you might need at least 100 rare diseases to check whether or not that linkage disequilibrium is seen, largely associated with the disease, is largely associated with the genotype correlation. And then you can come up with a diseasome approach. Then you can come up with a phenotype-genotype correlation, rather genotype-phenotype correlation. You will then ask, what is the disease phenotype, whether or not any disease phenotype has got a match with the genotype, and then go back and then ask that particular question again. Thank you.
Okay, thank you. I think you already answered from the omics point of view as well, right?
Yeah, I did that.
Okay, great. Okay, so we have another question from our beloved pengarah INBIOSIS, Prof. Zati. So her question is the definition of rare disease refers to the global scenario, or is it geographically related, or does it follow the international classification of diseases by the WHO?
Yeah, there is ICD-10 as well, and the World Health Organization. And there are 2 or 3 rare disease consortium as well, but they're also scratching their head on how to define a kind of a rare disease, unfortunately. Although in principle, the metric says that 1 in 100,000 could be a rare disease. But now that comes a question, whereas 1 in 100,000, say in maybe in Malaysia, that seems to be okay. But 1 in 100,000 in India, it may not be okay. Okay, or even in for that matter, China, it may not really be okay. So that kind of things are really geographically related. So each country has got its own this thing, as shown you in the very first few slides. And of course, the International Consortium for Rare Diseases, they are looking into these particular things. They are coming up slowly with the whole the World Health Organization, criteria as well. Thank you.
Okay. All right. I think I have another question from my side. I would like to know about the pipeline you shared just now, Sherlock, for identifying the variants. So actually, does it being introduced from your group?
Yeah, I mean, these are all the open source pipelines, I would say. I'm an open source person. I seldom use the commercial pipelines. So these are all the open source tools, but we tweak those particular tools. We benchmark the pipelines. We develop and we use different settings, and we try to modify it. So you simply give your query, a FASTQ file, you give it, and automatically it will give you the final list of mutations that you want to identify from the final list. Yeah, and then we simply take it, and that's what we are trying to standardize it in every year. My group is doing that, so we are doing that.
Great to hear that. So I would like to know, has the results from the generated from the pipeline has been validated from wet lab experiments?
Yes, very much. We do a Sanger validation in case of whole exome sequencing. We also done the genome-based approaches as well, as I was showing you. Every mutation, usually for all the rare diseases, of course, Sanger validation is a must. But in a rarity, in case of a DB sample that we have here, here we are still doing that Sanger validation right now. But the challenge here is again twofold. Even though we validate whether or not the mutation is sitting in the sample, we don't have a sample coming from either the parent. So we cannot say that even if the gene is monogenic, even if the disease is monogenic, even if we find those particular mutations, we cannot conclusively say that it's an autosomal dominant condition. Okay? So that's a kind of an inherent challenge that we are currently facing.
See. Do we have any more questions from the floor? Okay, another last question from me. Could you share your experience regarding the 3 Cs you shared just now? How to initiate collaboration with other fields? Because I can see that there are so much persons that.
Thank you for asking this, Rani. I mean, I really appreciate that. I mean, I always believed in openness. I mean, let's not hide any feelings within us. Okay. Suppose sometimes it might bring a lot of mount stupidity, but sometimes there's nothing wrong in showing our anger if the anger comes for a good reason. Yes. So say for example, I might be here, I might see something wrong happening over there in research. Maybe one particular paper is absolutely plagiarized. Okay, and I want to tell that particular thing in 2 ways. Maybe I can tell in one way. One person to one person, I might sound pretty harsh, but in bringing that harshness, I can bring him to the room, to my office, and say that yes, hey, look, you have completely plagiarized this particular article. This is not a good spirit that you really want to make it, for a good science that you're trying to do that. Okay, and then I tell him in a very open, in a very closed room like this, in a very open way, so that he'll try to modify those particular things. Likewise, if I really want to collaborate with anyone, with any persona, let's always keep that kind of openness. Let's not keep any kind of a barrier of keeping that kind of competitive inhibition, that so-called conflict of interest, or so-called competing interest. Even if there is a competing interest, so let's assume that I might be working on a rare disease, other person is working on a rare disease. Maybe we sit with each other. I don't see him as a competitor. In no means, in no way, he can probably do better than me, or maybe I can do better than him. We should only think in such a way that why not join hands together? Why not come and ensure that 2 brains come together, so that we can largely exploit that kind of collaborative spirit. So there's nothing wrong. I always write that particular mail saying that yes, I'm also working on this. Will you please try to collaborate? Okay, and suddenly then we agree on these particular things and we discuss this. That's how it is. And the second most important thing is, although we talk about the 3 C's, we should also talk about a E word. E word is expectant word, which is like what we say in BLAST. Okay, in BLAST also we use something what we call E value or expect value. Okay, so let's not keep any expectations. You do your duty. you do your task, you do your good thing, for excellent science that you want to do. You try to collaborate, but don't keep any expectations that your collaborations will really bring, this much amount of, energy, or maybe, this much amount of things. So just leave rest of the things, to the Almighty, to the God, and say that yes, you have done your justice, and do that. So the openness would always bring collaborativeness, and if the collaborativeness is always there, then there should not be any expectations. So this is what we call it as, the OC rule, the openness, collaborativeness, and expectations.
Okay, Dr. Prashanth, thank you for your answer, for your very excellent talk just now. And the take-home message for today is openness is key, right? For collaborative work. Okay, so I think we have come to the end of the webinar. I would like to thank Dr. Prashanth for the very interesting research topic, and I'm sure has opened up our view and give you an idea on how you want to bring up your research output to the next level. So today's webinar has recorded 38 participants on Cisco, WebEx, and 25 participants on the FB live page INBIOSIS UKM, making the total of 63 participants for today's talk. I would like to thank all participants for spending your time with us today.
Thank you. Thank you very much.
Thank you very much.
Yes, Sarna. Thank you.
Okay, wait for a while. Shatila, can you play the... Okay, so now I am delighted to share with you our service provided at INBIOSIS Center of Omics Data Analysis that we call CODA. You can use any dataset extracted from various omics platforms that include genomics, transcriptomics, proteomics, and metabolomics to produce publication-ready results provided by our experts here at INBIOSIS. And here are some of the publications that have been published previously for your reference. You can visit our website for more information of the service provided by CODA. Okay, now before we say goodbye, let us immortalize this session by switching on your video and have our faces captured. So technical team, please.